Welcome back friends. Welcome back to another video session from this channel. So in this video session we will be studying regarding the specific mechanism, the specific process which is involved in organizing the tRNA processing. Is it clear? So what is tRNA processing and what are the various modifications which successfully take place in the structure of the tRNA during the processing mechanism? So here, prior to the explanation of tRNA processing in detail, at first let us recall the structure of the tRNA. So what is tRNA? tRNA, likewise the other RNAs like rRNA and mRNA, it successfully synthesizes during the transcription mechanism by taking the advantage of an enzyme which is nothing but RNA polymerase 3. Is it clear? So let me write it on the board. So here tRNA is synthesized by RNA polymerase 3 So tRNA, it is the second most abundant RNA in a living cell, followed by rRNA. The most abundant RNA in a living cell is nothing but what? rRNA. After the rRNA, the next most abundant RNA in a living cell is nothing but what? tRNA. So let me write it here. It is the second most abundant RNA in a living cell. So tRNA, likewise the other RNAs like rRNA and mRNA, it also possesses or it also consists of certain usual bases like AUGC. Is it clear? So let me write it here. tRNA consists of usual bases like AUGC. So here if we consider the structure of the tRNA, basically the tRNA structure, it is composed of around 70 to 90 nucleotides in length. Is it clear? So let me write it here. tRNA is composed of 70 to 90 nucleotides in length. Is it clear? So here in order to elucidate the structure of the tRNA, various models, they were successfully explained by various molecular biologists. Out of these many models, the most widely accepted model of the structure of the tRNA, it is nothing but what? Cloverleaf model, which was explained by Hawley in mid 1950s. Is it clear? So let me write it here. Cloverleaf model it is the most widely accepted model of the tRNA. So here in order to organize the translation mechanism or the protein synthesis in case of prokaryotes and as well as in case of eukaryotes, here tRNA plays an important role. Is it clear? So here, as we all know, each and every amino acid which do exist in nature, it is very specific to one particular codon in the mRNA. But the amino acid itself, it is unable to recognize the codon which is present in case of the mRNA. So here, in order to recognize the codon which is present in case of the mRNA, the amino acid, it takes the advantage of a specific adapter RNA which is nothing but what? tRNA. Is it clear? So let me write it on the board. So here tRNA is involved in protein synthesis. So in case of prokaryotes, in order to organize the protein synthesis, there is an availability of around 30 to 40 tRNAs. But whereas in case of eukaryotes, in order to recognize or in order to organize the protein synthesis, there is an availability of around 40 to 50 tRNAs. Is it clear? That means each and every amino acid it is capable to encode with more than one particular tRNA. So the reason why in order to encode with 20 amino acids there is an availability of around 30 to 40 tRNAs both in case of the prokaryotic translation mechanism and as well as in case of the eukaryotic translation mechanism. Is it clear? So if we consider the structure of the tRNA, as I have already mentioned you, the tRNA structure basically it consists of certain usual bases like AUGC. In addition to the usual bases, the tRNA structure it also possesses or it also consists of certain unusual bases. Is it clear? So what are the certain unusual bases we are going to study then after? But prior to that, prior to the explanation of the unusual bases which are a part of the tRNA structure, at first let us have a basic look regarding the various sites which are available in the tRNA structure. So what are the various sites which are a part of the tRNA structure here? The various sites of the tRNA structure include acceptor stem. Is it clear? So here it is nothing but what? Acceptor stem. So what is the role of acceptor stem? As I have already mentioned you, each and every amino acid which do exist in nature, it is very specific to one particular codon in the mRNA. But the amino acid itself, it is unable to recognize the codon in case of the mRNA. So here, in order to encode the codon which is present in case of the mRNA, the amino acid, it takes the advantage of what? tRNA. So here the amino acid it successfully recruits to the tRNA at a specialized site which is nothing but what? Acceptor stem. Is it clear? So in addition to the acceptor stem, the tRNA structure it also possesses or it also consists of anticodon loop. So what is the role of anticodon loop? So as the name itself suggests, anticodon loop, that means the codon of the mRNA it is quite complementary to the anticodon of the tRNA. In order to encode the codon which is present in case of the mRNA, the anticodon of the tRNA, it plays an important role. Is it clear? So all these are the various specialized sites which are a part of the tRNA structure. So if we consider the various modifications which successfully take place during the tRNA processing mechanism, so the various modifications include chemical modifications. Is it clear? Chemical modifications, cleavage and trimming. So all these are the various modifications which successfully take place in the structure of the tRNA during the tRNA processing mechanism. Is it clear? So let me write it on the board. So what are those? Chemical modifications. Chemical modifications. And the next one is cleavage. And the next strategy it is nothing but what? Trimming. Is it clear? So at first we will be studying regarding the chemical modifications which successfully take place in the structure of the tRNA during the tRNA processing mechanism. Is it clear? So let me write it on the board. So if we consider the various chemical modifications which successfully occur in the structure of the tRNA include chemical modifications, right? So the various chemical modifications include methylation, is it clear? Methylation, isomerization and deamination. So let me write it on the board. Methylation, isomerization And the next modification is nothing but what? Deamination. So here at first we will be studying regarding methylation. So what is methylation? As the name itself suggests, during the methylation, the methyl group it successfully recruits to the nitrogenous base. So if we consider the guanosine and as well as cytosine. So here when the guanosine, when it undergoes methylation, under such condition, it leads to the formation of what? Methyl guanosine. Is it clear? In the same way, if we consider the cytosine nucleotide, when the cytosine nucleotide, when it undergoes methylation, it leads to the formation of what? Methyl cytosine. Methyl cytosine. Methyl cytosine. In the same way, if we consider the adenosine nucleotide, so here once when the adenosine nucleotide, when it undergoes deamination, is it clear? When the adenosine nucleotide, when it undergoes deamination, it leads to the formation of what? Inosine. So let me write it here. Inosine. In the same way, if we consider the uracil nucleotide, basically it is a nitrogenous base, right? So once when the uracil nucleotide, when it remains in association with the ribose sugar, it leads to the formation of what? Uridine. So once when the uridine nucleotide, when it undergoes isomerization, it leads to the formation of what? Pseudouridine and dihydrouridine. Is it clear? Pseudouridine and the other one is dihydrouridine. Is it clear? So during the transcription mechanism, by taking the advantage of RNA polymerase 3, the specific tRNA basically it consists of normal usual bases like AUGC. But then after, once when the tRNA, when it undergoes processing mechanism or the processing event, under such condition, these usual bases like AUGC, they undergo certain chemical modifications and they are nothing but what? Methylation, isomerization and deamination. So here you may get a small clarification why the particular tRNA which possesses or which consists of usual bases like AUGC, it undergoes various chemical modifications. So here the answer to this question is very simple. As we all know, the particular tRNA, it successfully synthesizes during the transcription mechanism in the nucleus. So here after the successful synthesis of the tRNA during the transcription mechanism by taking the advantage of RNA polymerase 3. Now the particular tRNA, it has to export from the nucleus to the cytoplasm in order to participate in the translation mechanism. So here the particular tRNA, it is a free floating nucleotide sequence in the cytoplasm. So as we all know, within the cytoplasm there is an availability of huge number of exonucleases, and these exonucleases, they are capable to cleave the extreme ends of the particular tRNA. So here in order to overcome the problem, the particular tRNA which possesses or which consists of usual bases, they undergo certain chemical modifications. Is it clear? So once when the particular tRNA which possesses or which consists of usual bases, when they undergo chemical modifications, under such condition, these exonucleases, they are unable to cleave the extreme ends of the tRNA structure. So the reason why the particular tRNA which possesses or which consists of usual bases, so generally they undergo certain chemical modifications like methylation, isomerization and deamination. Is it clear? So here this is regarding the chemical modifications. So then after we will be studying regarding the cleavage. So what exactly happens during the cleavage? So for your better understanding, let me present it on the board. And if we consider the next specific modification which successfully take place or which successfully occurs in the structure of the tRNA during the tRNA processing mechanism include cleavage. Is it clear? So why the particular tRNA structure it takes the advantage of cleavage during the tRNA processing mechanism. So for your better understanding, let me write it on the board. So here, so what it is? Cleavage. So as we all know, the particular tRNA, it successfully synthesizes during the transcription mechanism by taking the advantage of RNA polymerase 3. In addition to the common nucleotides, the particular tRNA structure, it also synthesizes certain extra nucleotides which are present at the extreme 5' and as well as at the extreme 3' end. So here in order to cleave the extra nucleotides which were successfully synthesized during the transcription mechanism by taking the advantage of RNA polymerase 3. The particular tRNA processing mechanism, it takes the advantage of certain exonucleases and as well as endonucleases. And these exonucleases and endonucleases, they play a prominent role in order to cleave the extra nucleotides which were successfully synthesized at the extreme 5' end and as well as at the extreme 3' end. Is it clear? So here in order to cleave the extra nucleotides which were present at the extreme 5' end of the tRNA structure, the particular tRNA processing mechanism, it takes the advantage of an endonuclease which is nothing but RNase P. Is it clear? So let me write it on the board. So what it is? RNase P. So here RNase P, it is the endonuclease which plays a prominent role for the successful cleavage of the extra nucleotides which were present at the extreme 5' end of the tRNA structure. Is it clear? So let me arrange the things here. So here the RNase P, it successfully cleaves the particular extra nucleotides exactly at this particular position. So let me arrange the things here. So after the successful cleaving of the extra nucleotides by the RNase P, then after here the particular tRNA processing mechanism, it takes the advantage of some more other endonucleases like RNase E and RNase F. RNase E and RNase F. Is it clear? So here RNase E and RNase F, they play a prominent role for the successful cleaving of the extra nucleotides which were successfully synthesized at the extreme 3' end of the tRNA structure. Is it clear? So let me arrange the things here. RNase E and RNase F. So these are the endonucleases which play a prominent role for the successful cleaving of the extra nucleotides which were present at the 3' end. Is it clear? So here if we consider the particular tRNA structure, likewise the rRNA and mRNA, even the particular tRNA, it possesses the non-coding sequences are the introns. So here the non-coding sequences are the introns which are a part of the tRNA structure. They won't undergo splicing by taking the advantage of transesterification reaction. So if we recall the rRNA processing and as well as the mRNA processing, in general the non-coding sequences are the introns. They are successfully spliced out by taking the advantage of transesterification reaction. But here the non-coding sequence are the intron which is a part of the tRNA structure. It won't undergo splicing by taking the advantage of transesterification reaction. So instead of taking the advantage of transesterification reaction, here the particular tRNA processing mechanism, it takes the advantage of an endonuclease. Cleaving of the tRNA structure by taking the advantage of various endonucleases. Then after the tRNA processing mechanism, it takes the advantage of one more other endonuclease enzyme. So here what is the role of the endonuclease enzyme here? If we consider the rRNA processing and as well as mRNA processing, The non-coding sequences are the introns which are a part of the rRNA and as well as mRNA. They undergo successful splicing by taking the advantage of transhistorification reactions. But here, in case of the tRNA, the non-coding sequence are the intron which is a part of the tRNA. It won't undergo splicing by taking the advantage of transhistorification reaction. Instead of taking the advantage of transhistorification reaction, the non-coding sequence or the intron, it successfully splices out by taking the advantage of a specific enzyme which is nothing but what? Endonuclease. Is it clear? So let me write it on the board. So what it is? Endonuclease. So here the endonuclease enzyme, it successfully cleaves at the 5' end and as well as at the 3' end. So once when the endonuclease enzyme, when it successfully cleaves at the 5' end, under such condition it generates or it induces an unusual hydroxyl group. Is it clear? 5' hydroxyl group. So in general at the 5' end, there is an availability of what? Phosphate moiety. But instead of having the phosphate moiety, here there is an availability of what? Hydroxyl group during the endonuclease cleavage. In the same way, at the 3' end, instead of having the hydroxyl group, here there is an availability of what? 2,3' cyclic phosphate moiety. Is it clear? So let me write it here. What it is? 2,3' cyclic phosphate. So in order to replace the hydroxyl group with the phosphate moiety, the particular tRNA processing mechanism, it takes the advantage of a specific enzyme which is nothing but what? Polynucleotide kinase. So let me write it here. Polynucleotide kinase. So as we all know, kinases are the specific enzymes which are most commonly responsible for inducing the phosphate moiety. So here the specific polynucleotide kinase, it successfully cleaves the unusual hydroxyl group which is in association with the 5' terminus, and here it replaces with what? The phosphate moiety. Is it clear? In the same way, in order to replace the 2,3' cyclic phosphate moiety, the particular tRNA processing mechanism, it takes the advantage of a specific enzyme which is nothing but what? Phosphodiesterase. Phosphodiesterase. So here the phosphodiesterase enzyme, it is responsible for the successful modification of the 2,3' cyclic phosphate moiety with the hydroxyl group. So let me write it here. Three prime hydroxyl group. So once when the usual groups, once when they were successfully recruited by taking the advantage of certain enzymes like polynucleotide kinase and as well as phosphodiesterase, then after one more other enzyme which is nothing but ligase, it comes into the picture. So what is the role of ligase here? Ligase, it is the specific enzyme which is responsible for inducing the ligation. Is it clear? Ligase, it is responsible for inducing the ligation. Ligation. Then after, the next specific modification which is a part of the tRNA processing, it is nothing but what? Trimming. So what is trimming here? Trimming. So here the tRNA processing mechanism, it not only takes the advantage of certain endonucleases like RNase P and RNase E and RNase F. And in addition, it also takes the advantage of a specific enzyme, which is nothing but what? RNase D, which is an exonuclease. So let me write it here. So what it is? RNase D. So what is the role of RNase D here? RNase D, it is the specific enzyme which acts like an exonuclease, and even it is responsible for inducing the trimming. So once when the 3' terminus, when it successfully trims out by taking the advantage of RNase D, under such condition, a specific enzyme which is nothing but what? Terminal nucleotide transferase enzyme, it comes into the picture. What it is? So let me write it here. Terminal nucleotide transferase. Nucleotide transferase. So what is the role of terminal nucleotide transferase? So it is the specific enzyme which is responsible for the successful recruitment of the trinucleotide sequence at the extreme 3' end of the tRNA. Is it clear? So all the various nucleotides which are a part of the tRNA, they are encoded in the tRNA genome. But whereas the trinucleotide sequence which is present at the extreme 3' end of the tRNA, it is not encoded in the tRNA genome. So here, during the tRNA processing mechanism, a specific enzyme which is nothing but terminal nucleotide transferase, it is the specific enzyme which is responsible for the successful recruitment of the trinucleotide sequence at the extreme 3' end of the tRNA. Is it clear? So let me write it here. So once when the non-coding sequences or the introns, when they are successfully spliced out by taking the advantage of certain endonucleases, and as well as once when the extra nucleotides, when they were successfully cleaved out by taking the advantage of certain endonucleases and exonucleases, and once when the trinucleotide sequence, when it is successfully recruited at the extreme 3' end of the tRNA, under such condition, the particular tRNA, it remains in mature conformation. Only the mature tRNA, it is capable to participate in the translation mechanism or in the translation process. Is it clear? So here this is regarding the specific mechanism, the specific process which is involved in organizing the tRNA processing mechanism. So here I hope that this video will help you a lot. So if you like this video, just hit the like button and share it to your friends. And I remember you people to subscribe my channel for getting more and more videos what I make day to day. Thank you.